We're going to discuss here today about the development of a project we have been working in the last 4 or 5 years. It's about the cholesterol interacting domain of TRKB as a new target for drug-induced plasticity. So essentially, we can boil down to the point that the question we want to answer here, in one of the questions we want to answer here at Customs Lab, is how do antidepressants work? Arrow has demonstrated since early 2000s that these drugs, they are able to activate TRKB receptor. TRKB receptor, as you probably know better than anyone else, is the receptor for BDNF. It's a neurotrophin receptor. It's a tyrosine kinase receptor, and antidepressants drug activate these receptor. The question is how? Because every time we try to interfere in this system, these drugs stop working. So our big question is trying to solve this black box concept that we have, that something happens that leads to an effect, to go to this is what happens, and try to learn something in the process here. So I'm not going to dig into the historical part of how these drugs do that, or when it was discovered. Arrow did that very nicely, summarized everything in this review here, published 3 years ago. So he tells the entire story on how they reach to that, what are the evidence, etc., etc., etc. What when I joined the lab in 2014, we were exactly trying to understand the deep mechanism. What are at the atomic or at the molecular level, what was happening when these drugs were administered to animals? And a lot of evidence was generated from the early 2000s to now that we know how these drugs don't do that. What are the many attempts were done to address these mechanisms, and mostly what we know now is how they don't do that. Until the point that in 2013 and 14, we decided to simply say, okay, let's not have any pre-hoc hypothesis. Let's get some animals, inject fluoxetine, which is the standard drug we use, pull down TRKB, extract the TRKB from the hippocampus, pull down, feed to a mass spectrometer, and see what comes up. And then we can start from there. So that's what was done. And to our surprise, nothing comes up. So not a single protein out of the 100 and something that we found to be part of TRKB complex, not a single molecule, not a single new molecule was there after fluoxetine. What we saw was actually less proteins were interacting with TRKB after fluoxetine. So fluoxetine was somehow disrupting the entire complex. At this point, we didn't know why, we didn't know how, but we start to dig a bit deeper into that, because one thing called our attention is it doesn't matter if you give the drug acutely to mice or if you give the drug repeatedly to mice, the same thing happens. And when you give a fluoxetine to cells, in this case here are fibroblasts that overexpress TRKB, the blockade of TRKB by K252A do not prevent this effect, this disruption effect. And anyone who ever tried to give BDNF and measure phosphorylation of TRKB knows that K252A is able to block it completely. However, we went one step forward, and what we found is if you simply lysate the cells and then you add the drug, that some of these interactions are lost. In this case, here's the example that I brought is for one of the components of the AP2 complex, which is responsible to internalize TRKB from the membrane. So this happens even without an intact cell. So it should be something fundamentally simple, something really molecule-molecule interaction. And then we found, well, sounds interesting. We had evidence that TRKB would directly interact with AP2M, is the Mi subunit of this entire complex. So maybe we're talking about that these drugs get into that interface and breaks it. So we designed a peptide with TRKB C terminal, we were able to complex this with AP2M, and then we added fluoxetine to this complex, and we didn't see anything happening. So this is what we see here. So this added to our list of this is not what is happening. But this stayed in our mind for a while. And we managed to publish this TRKAP2 story very nicely in the Journal of Biological Chemistry. However, still the bug was still there, how these drugs were doing that. And then we did what normally adults do. We crawl under our desks and we cried for a while. But then we decided to let's look to the structure of TRKB and see what goes on. And there is some literature that suggests an intrinsic relationship between TRKB and cholesterol. So they regulate each other. TRKB increases the synthesis of cholesterol, and cholesterol modulates signaling of TRKB. And then we also we linked that to one feature that is not present on TRKB, but in TRK-A and C, which was described by Eva Lembart in 2010. That is called this receptor dependence, meaning without the ligand, TRK-A and TRK-C leads to cell death. And this seems to be mediated by complexing with P75. But in a subsequent review from Bard's lab, they mention in the text that when they have the chimera of TRKB carrying TRK-A transmembrane domain, the TRKB behaves like TRK-A. So TRKB acquires this receptor dependence feature. Then we saw, okay, let's dig deep there. What is the difference between TRKB and TRK-A and C at the transmembrane level? And we found one thing that was particularly interesting. It's called is a motif, a recently described motif called cholesterol alignment and recognition consensus, or CRAC. In case of TRKB, we found is an inverted version of CRAC, it's called CARC, which is highly conserved, as conserved at least as TRKB. And the algorithm goes more or less like that. If you imagine the N to C terminal here, outside from inside the cell, at the membrane level, you need a central tyrosine flanked by leucine or valine in one side, and the other side flanked by an arginine or lysine. So you need this algorithm to work as a CARC if it's from the outside to the inside, or a classical CRAC, which is from the inside the membrane to inside the cell. And then we collaborate with a very talented group here in Helsinki. They did a molecular dynamic simulation, coarse-grained molecular dynamic simulation, and we found that the carbon for these two residues here, in the case of TRK, the tyrosine or the arginine, they are able to dock cholesterol. However, in the case of the central tyrosine, this interaction is 10 times higher than for the arginine. So it's okay. Now we finally have something to go to look for. And as I said, this domain is not found in TRK-A, transmembrane region. If you look here, nothing, or in TRK-C. So something exclusive of TRK-B. Then we got some fibroblasts that we have here. They overexpress TRK-B or they overexpress TRK-A. We have these two cell lines. And what we tested is cells expressing TRK-B on here, they respond to cholesterol, which is blocked by this cholesterol sequestering agent, this beta-cyclodextrin. And what cholesterol do seems to be to hold TRK-B a bit longer in the membrane, so you have an accumulation of TRK-B in the membrane. The surface TRK-B goes up, and nothing happens with TRK-A. So I said, okay, now we have a possible function for these domains. It interacts with the membrane and regulates the surface positioning of the receptor. Any functional role for that. So using culture of primary cortical cells from rat embryo, we observed that there is a bell-shaped curve for cholesterol, modulating the TRKB phosphorylation upon BDNF stimulation. So lower doses of cholesterol leads to favors TRKB phosphorylation induced by BDNF. Higher doses go back to the control levels. Even higher, you compromise this effect. So we have this nice bell-shaped curve for cholesterol. Then we thought, well, we have this central tyrosine that docks cholesterol apparently, and we have this bell-shaped curve. Any evidence that cholesterol would remotely bind there? So what we did, we used these microscale thermophoresis. Let's take a minute to go for the method here. It's a measure of the fluorescence of a target or a ligand. The changes in fluorescence that happens upon a temperature gradient. So you heat the sample, molecules start to move faster, and then you start to lose fluorescence. If you have a ligand there that potentially changes the conformation of the target, then this movement is compromised. So you see a delay on that. It's more or less what you see here in these representative plots. So you turn on the laser, you start to lose the fluorescence, and then upon the ligand, you start to have delayed loss of this fluorescence. Okay, so very, very interesting, very nice technique. And then we got TRKB. We label it with GFP. Then we have our label target here, and we start to add cholesterol on that. And we indeed saw, okay, seems that cholesterol interacts with TRKB, but when we make this mutation, this central tyrosine to phenylalanine, and we made this mutation because, remember, tyrosine, this central tyrosine is inside the membrane, and in transmembrane domain is not something you can mess around very freely. So we have to change carefully to choose carefully what we want to mutate. But keep in mind that this mutation here completely abrogates cholesterol interaction with TRKB is one oxygen off. It's a tyrosine, which is hydroxy, to a phenylalanine that doesn't have any hydroxy, but just the hydrogen there. Then we went back to the molecular dynamic simulation, in this case with a more refined method, it's called atomistic simulations. And what we observed is that apparently on top of interacting with TRKB, what cholesterol do is regulate the membrane thickness, which you can see happening here. The more cholesterol you add, you change the conformation of the dimer of TRKB. So TRKB, once dimerized through this AXXG central motif here, is well known for dimerization, you achieve a certain distance or a certain angle between the intracellular or the extracellular portions. And upon cholesterol, tyrosine works more or less as a floating device. So the hydroxy will always be on the top or the edge of the membrane. So the more thick the membrane goes, you start to change the orientation of TRKB of the monomers related to each other. And then, according to their calculations and according to similarities with what we know about the EGF receptor, there is an optimal angle here that is determined by the thickness of the membrane. More than that, you start to twist your transmembrane domains. And what we observed is that even at this optimal, let's call this Goldilocks zone for the angle between the monomers, when you have a mutation, when you have the tyrosine phenylalanine mutation, one of the monomers rotate related to each other, which potentially can compromise the signal. Then we said, okay, sounds interesting. Let's see that in action. Let's see if this changes the TRKB function somehow. And on that, we used a method called Fluorescence Recovery after Photobleaching, or FRAP, where we can get hippocampal neurons, transfect them to express the same GFP-labeled TRKB. And with a high-power laser, we can go to the tip of the spine and bleach it, and then we count the time that takes for the fluorescence to recover, to go back to the basal levels. And what we found is that in the shaft, You can look here in these first three pictures. In the shaft, the fluorescence goes back to normal, but nothing happens in the spine. This is the same neurite. So the spine is a very, very stuck structure. However, when we add fluoxetine, we see a recovery in the spine. You can see here going up. And what was interesting, if you add ketamine, you also see that. You also see this recovery going on. But in the mutant TRKB, we see nothing. Nothing happens. Those go, doesn't matter if you add the drug in the system. Then we thought, well, okay, we have cholesterol interacting with this domain. We have evidence from Mark Razenik's from lab from Chicago that have been describing the accumulation of these drugs, many antidepressants, including ketamine more recently, and ketamine metabolites in lipid rafts, which are cholesterol-enriched domains in the cell membrane, and especially in the spine, which is particularly rich in these rafts. And there's a lot of debate about the function of the lipid rafts, but they would serve as signaling platforms. And then our next question was, well, we have cholesterol interacting there. We have these drugs accumulating in the rafts. Would they be binding somehow to this TRKB, to this TRKB dimer? Then we went again back to our molecular dynamic simulations, and we start some docking experiments where we got that Goldilocks conformation of TRKB, and we start to stuck fluoxetine around it and see what if it docks, if it stays there. And what was very interesting, from at least from our point of view, is that fluoxetine seems to interact, at least for the molecular dynamic simulations, seems to interact with TRKB, but many criteria has to be fulfilled. One of them is it needs to be the dimer, which is alone is already a new information. We normally think that a ligand and a receptor would work as one-to-one. In this case, you need two molecules of TRKB for one molecule of fluoxetine, because you imagine TRKB is acting like a Caesar here, and what fluoxetine does is stuck and differentially interact between the two monomers. This is one of the first. This was the first interesting data we got from the molecular dynamics for the docking simulation. The second one, as I said, is you need TRKB to be in this right orientation. Okay, it doesn't. If you stuck fluoxetine in any conformation of TRKB, it's not going to last long. But in this one, in this right orientation, you get it. The third important feature, the third requirement is you need the membrane environment there, because phosphatidylcholine and other membrane lipids, they act as a lock to this whole complex. And then we have to rethink many things. So the binding is not depending just in the simple interaction of TRKB and fluoxetine. It also needs the environment matters a lot. We found, okay, this is awesome. It's a very interesting result. Do we have any evidence from that wet lab evidence? And then we designed a simple binding assay. Luckily, some drugs you can bioaccumulate, no problem, especially primary and secondary aminos. Fluoxetine is one of that. We can bioaccumulate fluoxetine, and then we pull down TRKB and add labeled fluoxetine to that. And what we observed is that there is a low affinity interaction, at least, between fluoxetine and TRKB, which is severely decreased in the mutant. Another drug that we can bioaccumulate is hydroxynorketamine, and this is one of the active metabolites of ketamine. And we saw more or less the same profile. So there is a low affinity binding site, low affinity interaction, at least, between HNK and TRKB, which seems to be compromised by the mutation. Another drug that we cannot bioaccumulate very easily is imipramine, but we can buy some tritiated imipramine. And we saw exactly the same profile. It interacts at a micromolar affinity range, and the interaction is compromised by the mutation. Unfortunately, ketamine is not one of the drugs you can bioaccumulate easily, but what you can do is compete fluoxetine out. And that's what we did. We start to add cold as ketamine versus hot fluoxetine, and we saw this nice decrease. Again, with the IC50 going around the revolving around 1 micromolar zone. Next thing we did, well, these drugs are accumulating in the rafts. They are interactive TRKB, more or less similar to cholesterol. So are they cholesterol mimetics? And then okay, let's compete fluoxetine out with cholesterol. And nature had other plans. No, they don't act as cholesterol mimetic. Apparently, what they do is they need cholesterol to interact, because when we add cholesterol to the story, you can see here, we see a favoring of this interaction. And this is another interesting data that we have. And we also saw that the same thing happening with HNK. But nothing changes with BDNF here. BDNF, the mutation does not alter BDNF ability to interact with TRKB, or BDNF also is not able to dislocate fluoxetine, meaning they are not competing from the same side. And here we did again the microscale thermophoresis just to look at the same, try to look at the same evidence from a different method. And that's what we observed here. We got a nice interaction between fluoxetine and that GFP labeled TRKB. Okay, that's a nice amount of in vitro evidence for that. Any relevance, any behavioral, any system relevance in this for the mutation. At the time, then we were very lucky because we were able to CRISPR this mutation. It's a single amino acid, so it's pretty much very CRISPRable, you could say. So, and the animals are viable. The animals are fine. They just carry the mutation. They're not double mutants. And then we got the animals, and it's okay. Now we have a Disneyland to play on. We have many models that we could start to play. One of the first that we did is the shift in ocular dominance induced by antidepressants. And because Arrow has demonstrated about more or less 10 years ago that fluoxetine in combination of monocular deprivation switches the ocular dominance in the adult visual cortex. And adult visual cortex is not something very plastic. It's something that's very plastic during the critical period when the animals are young, but once the connections are established, it is very hard to change it. And it's a gold standard model of plasticity. So what we did is we got these animals, we made an image of their monocular dominance upon stimulation. We put a cranial window on them. We give a visual stimulus, and then we can measure the bold signal that we get in the primary visual cortex. And then we can make this before and after the drug treatment that we see here. Fluoxetine, and we have recently published the data about that, you need 3 to 4 weeks to work. And so we treat the animals for 4 weeks. In the last 8, we monocularly deprive them. And that's what we see here, is that fluoxetine induces a shift in ocular dominance. This was demonstrated before. This is associated with increasing BDNF in the visual cortex, but when you look to the mutant animals, nothing happens. Another drug that we wanted to test is HNK, which is also seems to work in this model, but it works very quickly. Sorry, but this little bar up here, you can see HNK, but we did only 3 injections alongside these 8 days of molecular deprivation. And what we saw is, yeah, HNK changes, makes the switch, so decreases the ocular dominance, and the animals carrying the TRKB mutation are resistant to the treatment. Next model we used, which was object location memory, which is a variant of NOR, of novel object recognition, where we have 2 identical objects in an arena with cues for the animals to localize these objects. And then we train them for few days with these objects. They are completely irrelevant. They are neutral objects. And then we treat them, and on the test day, we move one of the objects to a different location. What animals, especially mice, normally do, they're very curious. They start to interact more with the new object, newly located objects, not a new object, a newly located one. And then we observed that fluoxetine improves this performance if started after the training session. And again, TRKB YF mutants are resistant to this treatment. It's the same way as BDNF animals. BDNF hat animals are also resistant to that. It's a nice control that we have to add to this model. Third model that we tried is fear conditioning, extinction of fear conditioning. Again, we demonstrate some time ago that fluoxetine in combination with the extinction training facilitates the extinction, meaning you decrease the freezing of a previously conditioned tone, or in this case, to the previously conditioned context. And what we saw, again, fluoxetine decreases the freezing, but nothing happens with the mutant animals. And same thing happens to ketamine. Ketamine decreases the freezing, but nothing happens with the mutant animals. Then the last behavioral approach we did was actually the first one we should have been done in the beginning, which is the classical model to screen for antidepressants, which is the forced swimming test. And then this is the no need for explanation, so everybody knows or have seen the forced swimming test, but it's pretty simple. You put an animal in a cylinder and then you count the time it takes, it stays immobile in a given time. There are many variations of that, but let's not go into the details. And what we found, as for the other three models we tested, Y-type animals respond both to fluoxetine and to ketamine, but the mutants are, again, resistant to this drug. So from this point, we start to think about what is the model we want to use to describe that. So we start to think about at the molecular level that would reflect in what's happening at the molecular level that would reflect in behavior. And that's what we came up. This is the framework we came up. In physiological conditions, TRKB would oscillate between a signaling competent form to signaling incompetent forms, depending on the membrane thickness, which will be determined by cholesterol. So at higher levels, nothing happens. At low levels, nothing happens. So you need, again, the term, the Goldilocks zone. And what fluoxetine and apparently other drugs do, they stuck there and hold TRKB in the signaling competent form, even at higher concentrations. So you maintain TRKB a bit longer in this conformation. And this is more or less what is described here for the interaction of the drug when you stuck the drug there in this wedge, and you count the time it takes to leave it. This is again a simulation. And after few nanoseconds, a couple of hundreds of nanoseconds, you already lose completely the drug. But this is much faster in the TRKB mutants. It doesn't seem much. You go from 300 nanoseconds to 600 nanoseconds, 300 nanoseconds, super fast. But imagine that. You have millions of TRKB molecules, millions of dimers, and each one of them, each pair is few nanoseconds longer signaling. So when you multiply that, when you start to look at what's going at the synaptic level, makes sense to think that, well, this teeny tiny extra time is enough to at the end culminate in a higher signaling effect. And if everything we have been observed for the role of TRKB, BDNF TRKB system for antidepressant effect seems to be crucial for that. So that's now the state that we are. We are trying to refine that, and let's see what the future goes. And with that, I finished my presentation super quick than I was expecting. I just have to thank, of course, to our funders. We have been very well funded during these last years. To our collaborators, Mart Sarma and Vera Kovaleva, who did the microscale thermophoresis, Ilpo Vatulainen and his group, who did all the bioinformatics approach, Clive Brahman from University of Bergen, and Klaus Norman, also from Klaus Norman from University of Freiburg, for collaborating with the electrophysiology analysis. Some data that I didn't show here, but they help us a lot to build a nice story. To everybody in our group, especially Eero and all the students that have been actually did the heavy lifting or conducting experiments, and especially for Carol, who helped a lot develop this idea. I will use the last slide to make a shameless advertising. We recently launched a journal to focus exclusively in publishing replication results in neuroscience. This is backed by the University of Helsinki, and it's completely and totally edited and maintained by us, by academics. So there we can operate in a diamond open access model, meaning there's no APC, there's no fee whatsoever to publish or to read. We are focused in publishing attempts to replicate results. So if you ever try that, and it's well conducted, it's well designed, send to us.